A very good morning to one and all. On the behalf of the St. John's Institute of the Pharmacy and the Research, I, Samiksha Mhatre, a student of MPharm in Quality Assurance, extend a warm welcome to each one of you to SCIENTIA, a knowledge sharing session by an SJIPR faculty, and introduce you to an college campus by sharing a small video. Now, I would like to begin with our 27th session of SCIENTIA today, which is entitled as Biosimilars, and it gives enough pleasure to introduce our speaker to today's session, Macveena Machado ma'am has been working as an assistant professor in Department of Pharmaceutics in St. John's Institute of Pharmacy and Research, Palghar. She has completed her B Pharmacy from St. John's Institute of Pharmacy and Research, Palghar in the year 2016. She has completed M Pharm specialization in quality assurance from Vivekanand Education Society College of Pharmacy, Chembur, Mumbai in the year 2018. She is having total 3 years of academic experience. Her key research areas are formulation and development of conventional and novel drug delivery systems, formulation and development of herbal and nutraceutical-based drug delivery systems. She is a life member of several professional bodies like life member of Indian Society of Technical Education, ISTE, life member of Indian Pharmaceutical Association (IPA), registered pharmacist under Maharashtra State Pharmacy Council (MSPC). With this brief introduction about ma'am, now I request ma'am to take over the session.
A very good morning to one and all present for this SCIENTIA session. The topic on which I'm going to talk about is biosimilars. Some of you all might have heard this term, but I'm pretty sure some of you all may be still unaware of this term. So I will be covering just the basic aspects of this topic, biosimilars. So the flow of this presentation is we are going to do the introduction to biologics and biosimilars. Then we will see what are the differences between biosimilars and generics. We will take a look on how these biosimilars are developed, that is development of biosimilars. We will see what are the approval requirements for the approval of biosimilars, and then a short summary. Now let us take a look at what are actually biologics and biosimilars. So all of y'all might have heard what are biologics, which are also known as biological medicines. From the name itself, you all might understand that biological medicines means any class of medicines which is derived, in which the active ingredient is derived from a living organism. And particularly this biological medicines, by no means it can be synthesized by chemical means. So biological medicines are those class of medicines in which the active ingredient comes from a living organism. So the official definition of biologic is provided by the US Federal Code of Regulations, that is CFR. And the definition of a biologic is it is any virus, therapeutic serum, toxin, antitoxin, or any analogous product applicable to the prevention, treatment, or cure of diseases or injuries of man. So this definition varies from country to country. Each country's regulatory agency provides a different definition of biologics. But the bottom line is the biologic, it basically includes a wide range of products which are available today as vaccines, blood and blood components, certain allergenics, somatic cells, gene therapies, tissues, and recombinant therapeutic proteins. So all the medicines that are derived from living organisms and which cannot be produced by chemical means come under the aegis of biological medicines. These biological medicines, they are highly specific and targeted medicines, and they are now an integral and indispensable part of modern medicine. So the biological therapies which are available today, they include different classes of medicines like enzymes, vaccines. Human insulin was probably the first biological therapy available to the humankind. Then interferons, interleukins, erythropoietins, gonadotropins. Even we have granulocyte colony stimulating factors, hormones, which the most famous category is human growth hormone. Then we have monoclonal antibodies, blood coagulation modifiers, and tissue plasminogen activators. So the biological therapies which are available worldwide today and which are currently in use, they belong to either one of these categories. So these biological drugs, since they are developed or since they are available from the living organisms, they are usually large molecules. And these molecules, they have tens of thousands of atoms which are present, making them bigger than the molecules that comprise our normal drugs. So in the image which is provided over here, you will see the structure of ibuprofen. And in comparison to ibuprofen, if you compare a biological medicine that is monoclonal antibody, the structure of this monoclonal antibody is far more greater than your normal chemically synthesized drug. Okay, so here in this slide, we will see what are the major differences structurally in your small molecule drug, that is your normal chemically synthesized molecules, the proteins, and then the monoclonal antibody, which is actually your biological medicine. So if we compare your small molecule drugs with your biological drugs, the structure is quite large for your biological medicine, then the small molecules which we deal with, they are usually a single molecule or a single substance. But if we go on to look at the biological medicine, they are a mixture of variants. Because they are synthesized biologically, they will include different types of amino acids, different types of processes like the post-translational modifications, which include, due to which mixture of variants is generated that will eventually increase the size of those molecules. The small molecules usually have a very low molecular weight, but if we compare biological medicines, they usually have larger molecular weights. Even if we look at the mechanism of action of small molecules, the mechanism of action is mostly ambiguous for your small molecule drugs. But if we see the mechanism of action of your biological medicines, they are very specific and target oriented. So these are some basic differences between your small molecule drugs and your biological medicines. So if we go on to a more detailed difference between the small molecule drugs and biological drugs, the differences are mostly related with product and manufacturing. So the small molecule drugs, they are produced by chemical synthesis, and biologic drugs, they are biotechnologically produced by host cell lines. Small molecule drugs, as I mentioned earlier, they have low molecular weight. Biologic drugs, they have high molecular weight. The small molecule drugs, they have very well-defined physicochemical properties. That is, if we define the physicochemical properties of any of your chemical moiety, we can properly state as to what are those physicochemical properties. But due to the complex nature or the complex structure of your biologic drugs, the physicochemical properties of those biologic drugs are also complex. Small molecule drugs are relatively stable, but if we see biologic drugs, they are very sensitive to heat and other external environmental conditions. The small molecule drugs, they are single entity or a single molecule, but biological drugs, they are heterogeneous mixtures, which are basically due to the complex manufacturing processes. Small molecule drugs, they can be administered in the body by different routes of administration, but biological drugs, they are usually administered parenterally. The small molecule drugs, they will rapidly enter or they will get rapidly absorbed even if we administer it by any routes of administration. But since biological drugs, they are large molecules or they are bigger in size, they reach circulation only via lymphatic system. And also during this absorption, they are subjected to proteolysis during interstitial and lymphatic transit. So the absorption of these biological drugs is also difficult to monitor. The small molecule drugs distribution, if we see, they can get distributed rapidly to any organ or tissue. But the distribution of biological drugs is usually limited to plasma and extracellular fluid. The small molecule drugs are non-antigenic, and the biological drugs are usually antigenic. Now, this point is very important to consider because the immunogenicity produced by small molecule drugs is negligible. That is, the small molecule drugs will not induce any reaction, immune reaction inside the body. But since biologic drugs, they are large in size, when they are administered inside the body, immune response will be triggered. Our body will consider these biological drugs as some foreign materials, and they will start developing an immune response towards it. And sometimes this immune response could be fatal in some instances. So the manufacturing differences for small molecule drugs and biological drugs, we know exactly how can we synthesize a small molecule drug. And these, after manufacturing, if we want to characterize or if we want to know the structure of that small molecule drug, we can easily find out the structure by analytical method. But for biological drugs, the manufacturing process is complex. And if we want to know the actual structure of such biological drugs, we do not have enough methods which will be able to characterize the entire molecule completely. Also, the small molecule drugs are easy to purify, but the biological drugs, they are lengthy and they have complex purification processes. The small molecule drugs, during manufacturing, the contamination can be avoided. And if contamination is present, we can easily remove that contamination. But for the manufacturing of biological drugs, there is a high possibility of contamination. And if contamination is present, the detection and removal is often impossible. For small molecule drugs, even if we make slight changes in the production process and environment, there will be no changes in the final product. But for biological drugs, these drugs are usually highly susceptible to slight changes in production process. And even if we slightly change the manufacturing process, the end product will have a great degree of variation. So these biologics, they are used from a long time, and they are used to treat many different life-threatening and chronic diseases, such as your diabetes, or mainly the chronic diseases, diabetes, blood-related diseases, inflammatory diseases, your gastrointestinal ulcers like Crohn's disease or inflammatory bowel syndrome, certain immunological or autoimmune diseases. So all such chronic conditions can be controlled properly by using biologics. Now, why did I provide you all an overview of biologics is that because biosimilars are related to biologics. So biosimilar medicines, or more commonly biosimilars, they are developed to be highly similar versions of approved biologics that have reached the end of their proprietary patents. Now, basically, biologics were first introduced somewhere in 1950s or 1960s. So during that time, the manufacturers or the innovators who produced biologics, they filed patents. But once the patent expired, other companies who wish to take up that particular biologic can do it. Okay, so whenever a different company, apart from the innovator company, takes up that same biologic and develops it or markets it, that biologic will get converted into biosimilars. So these biosimilar medicines, they are highly similar versions of approved biologics. So these biologics, they are known as originator biologics or reference medicinal products, which are used for the development of biosimilar medicines. Whereas biosimilar medicines are biologics which are marketed once the patents related to the originator biologic have expired. So same we have in our pharmaceuticals also. We have an innovator drug molecule. When the innovator drug molecule's patent expires, it gets converted into generic. So the same concept applies to biologics and biosimilars. Biologics are innovators and biosimilars are highly similar products of biologics. So the biologics or innovator biologics are used as a reference product for the development of biosimilars. So usually when we deal with biosimilar or biosimilarity, it means that the biological product is highly similar to the reference product, notwithstanding minor differences in clinically inactive components. So the components which are present in a biological product, the same components should be there in a biosimilar product. But as I will explain you all in the next slides, it is highly similar, but it is not identical. So when we compare biologics and biosimilars, they need to be highly similar, but they cannot be exactly identical to the innovator biologics. The second point of consideration in biosimilarity is there are no clinically meaningful differences between the biological product and the reference product. So the meaning of this no clinically meaningful differences means that the activity or the clinical activity of the biosimilar and the biologic should be completely equal. So even if the structure has some minor differences, still the clinical activity, clinically, both biosimilar and biologic should have the same activity. So again, I've already told you all what is the reference product. So when we refer the original branded and approved biological product in the context of biosimilar medicine, it is called as the reference product. Okay, so innovator biologics are usually the reference products which are used for the development of your biosimilars. So the term biosimilar is a very common term, but different agencies have different terminologies accepted for biosimilars. So biosimilars are known as follow-on biologics in Japan. WHO calls biosimilars as similar biotherapeutic product. In Canada, biosimilars are known as subsequent entry biologics, whereas India uses the term similar biologics. These are some official definitions of biosimilars which are provided by the European Medicines Agency, the FDA, and the World Health Organization. So according to the World Health Organization, a biosimilar is a biotherapeutic product which is similar in terms of quality, safety, and efficacy to an already licensed reference biotherapeutic product. So basically, the biologic or the biosimilar should be similar in terms of quality, safety, and efficacy to an already licensed reference biotherapeutic product, that is an innovator biologic product. So this is basically the difference between the biosimilar and a biologic. So the question that many of y'all might ask is, are biologics and biosimilars the same? They are identical, they have the same activity, but we cannot call them as same. So all biosimilars have the same amino acid sequence as their reference biologic. So the basic or the basis of their structural foundation is the same. But when biosimilars are developed or when biosimilars are manufactured, the manufacturing usually starts through cell lines wherein we do expression in the genes, and the process takes place biologically. So there is a complexity of the manufacturing process, and biologics, they have a high degree of variability between the different batches of the same product. Okay, so we cannot control all the process variables during the manufacturing of biologics and biosimilars, and that is why if we produce different batches of the same biologics or biosimilars, some degree of variability will be observed in the structures of those biosimilars. But we can control this natural variability in both biologics and biosimilars, so that we can ensure consistent safety and efficacy. So the degree of variation should be controlled. We cannot allow a high degree of variability which is present between the biologics and biosimilars, but some degree is acceptable provided they are consistent in safety and efficacy. So these images will explain you all the basic difference or the differences that arise in biologics and biosimilars. So the example of this biologics is infliximab, which is used mostly in Crohn's disease or inflammatory bowel disease. So the biosimilar to infliximab, which is developed, is infliximab DYYB. And you all can see the sequence or the structural difference which is present in this biologic and biosimilar. So this difference mainly arises because the process is completely biological, and during biological processes, due to some post-translational modifications maybe, there is a possibility that some structural variations can take place. So why should we use biosimilars? Biologics are already available, but why the concept of biosimilars has now come into practice? So there is more than 10 years worth of real world experience stating that biosimilar medicines offer the same effects to the patients and healthcare system as biologics. So the disadvantage of biologics is they are very costly. So the biological treatment that was provided, it has a very high cost. Plus the biologics which were patented previously, they are going to soon get expired. Okay, so patent usually for biologics is granted for a period of 20 years. So mostly it was seen that in the year 2017 to 19, majority of the patents which were filed were going to get expired. So during those 2 years, there was a hike in the production of biosimilars. Also, the chronic diseases are highly prevalent nowadays worldwide. So in order to control or in order to treat those chronic diseases, biologics or biosimilars, they are quite promising. And also they provide an alternative to stakeholders. So alternative treatment therapies are available both to the healthcare providers as well as to the patients. Okay, so here just an example that how long it takes for a reference biological development process and the total cost. So if we consider reference biological, it usually takes a lot of time, probably 10 to 20 years, and the total cost is greater than $1 billion. But if we compare the biosimilar development timeline, it usually takes some 7 to 8 years, and the total cost is also reduced tenfold. That is, the total cost for the development is 100 to 200 million dollars. So mainly because of the cost and the time required for development, now the shift has taken place from biologics to biosimilars. So this biosimilar medicines, they provide benefits to patients, healthcare professionals, as well as to the payers. So the patients, they gain more access to the more treatment options, and usually at earlier stages of the therapy cycle. So if a cancer patient wants to avail this biological treatment, and if he is short on the expenses, obviously he can go to biosimilar therapy, which is available at a very less cost. Also, it will improve access, which will give better outcomes for the patients. Also, the healthcare professionals will have wider options or treatment options which they can provide to the patients. They will develop value-added services for patients via benefit sharing model. So the benefits which are associated with biosimilars are great. So by sharing this experience of benefits of biosimilars, they can give value services to the patients. And also they will reduce pressure on the prescribers. Also the payers, that is mainly your marketing authorization holders, they will have a competitive market with a broader range of cost-effective treatment options. So when biologic patent is expiring, there will be other players who will be ready to produce biosimilars, and they will create a good competition due to which cost-effective treatment options will be available. And in turn, it will generate savings across healthcare systems, which will support their sustainability. So again, an example for the biosimilar medicines, which making biotherapeutics a cost-effective option and broadening treatment choice. So the cancer treatment, generally it induces anemia in the patients. So according to the 2008 NICE guidelines, epoetin is clinically effective for cancer treatment-induced anemia, but it was not cost-effective. So epoetin, it is a biologic, but it was not cost-effective. But again, after 6-7 years, the epoetin, which was developed as a biosimilar, it was both clinically effective and cost effective. So this biosimilar medicines, they empower physicians by providing cost-effective treatment options. Now, I have been telling you all about the generics which are available for your chemical drugs, and biosimilars, which is for your for your biologics. So basically, biosimilars are not the same as generic drugs. Generic drugs, they are usually produced or they are usually approved by the ANDA pathway. But the biosimilar product, they are approved by separate guidelines which are provided by the regulatory authorities. So if you see lot-to-lot variability of your generic drugs, there is no variation observed in the structure of your generic drug. But if we see the biosimilar product, there is lot-to-lot variability. Slight differences are seen in different lots of the same biosimilar product, which says that generic drugs and biosimilar products, they are not the same, they are different. So these are the differences between generic medicine and biosimilar medicine. Again, generic medicines are small molecules, biosimilar medicine is larger and structurally more complex. Characterization of generic medicine is easier. Biosimilar medicines characterization is very tough. It requires multiple technologies for their complete characterization. Generic medicines produced by chemical synthesis, biosimilar obtained from biological source. The generic medicines, after production, they get the same, exactly the same copies of the same molecule. But when we produce biosimilars, we cannot get the exact same copy of the biological. However, some variability is observed between the original biologic and the biosimilar. For generic medicine, the pre-approval characteristics which are required are clinical data requirements. That is mainly pharmacokinetic bioequivalence studies are required so that we can market generic medicine. But for biosimilar medicine, we have to perform comparative pharmacokinetic and pharmacodynamic studies. Safety and efficacy data may be required, particularly for more complex biological medicines. So for generic medicine, only if we perform bioequivalence studies that will do the purpose, serve the purpose. But for biosimilar medicine, we need to show whether the biosimilar medicine is equal in efficacy to your original biologic. So the development of biosimilars is quite interesting. Biosimilars are reverse engineered. Reverse engineering means that we know from which DNA or from which gene we will get that particular biosimilar. But we must develop a product that matches up to the reference product in virtually every way in terms of its physicochemical properties, pharmacokinetics, pharmacodynamics, immunogenic properties, and these are evaluated on each and every step of the biosimilar evaluation process. So we know basically what we are looking for, but at each step of this development process, we need to establish or we need to prove the similarity between the biosimilar and the biologic. So each step has critical ramifications in the drug development of biosimilars. So the reference product or the biological product process is quite simple. They select a cell line, they select a process, and they manufacture the biologic. After the biologic is manufactured, they demonstrate the safety and efficacy by performing clinical trials. They validate and control the manufacturing process, and they create a reference fingerprint or a reference product. However, if we look at the biosimilar process, the development of biosimilars starts with characterizing the reference fingerprint. So this is the reference product or the originator biologic. This reference fingerprint is taken by the biosimilar, the company who is developing the biosimilar. They will characterize the reference product. Then they will choose a cell line and they will choose the process. Now the process which was used by the innovator company is not disclosed to the biosimilar development company. So the biosimilar, the scientists who are developing this biosimilar, they will have to generate their own process. So once they characterize the reference product, they create a cell line, they develop a process, and then they go on to establishing biosimilarity. Okay, so biosimilarity is established by demonstrating the safety and efficacy. They will also have to perform the clinical studies on that biosimilar which is developed. And if enough evidence is generated that it can be a biosimilar candidate, then they go on to the actual manufacturing of the clinical product. But if biosimilarity is not established, then again they have to go back, characterize the reference product, create a different cell line, create a different process. So unless and until biosimilarity is not established, the biosimilar development process will not proceed. So after the biosimilar is being manufactured, we demonstrate comparable safety and efficacy. So basically we compare the biologic and biosimilar, and then we demonstrate whether they are comparable with respect to safety and efficacy or not. So once we demonstrate the safety and efficacy, then we can go on to the validation and establishing controls, and then we can give out the biosimilar specifications. So the stages of biosimilar development are, the first step is intensive physicochemical characterization. That is, we establish the sameness of the reference, that is the biologic and the biosimilar. That is, the biosimilar and the biologic should have identical amino acid sequence, and it should be indistinguishable by 3D structure. So the 3D structure of both biosimilar and biologic should be exactly the same. Then functional analysis to confirm the sameness of the observed comparative, analytical, and functional assays, which will demonstrate that there are no clinically relevant differences between the reference medicine and its biosimilar. So the functional analysis will determine whether there is a sameness in the clinical activity of the biologic and the biosimilar. Then they go for preclinical in vivo studies to demonstrate comparability at structural and functional level. Then they go for clinical phase 1 pharmacokinetic and pharmacodynamic studies, and then clinical phase 3 confirmatory study. So after these steps, they confirm the efficacy, safety, and immunogenicity of the biosimilar with the biologic. Now, basically, the biosimilar development follows a scheme of totality of evidence. Now, totality of evidence is actually a data package that is generated from the biosimilar development program. So one example I have attached as a snapshot over here. Now, if we want to determine the primary structure of the biosimilar, so what we do is, one such analytical study is magnetic resonance. So what we do, we take the spectra of reference medicine and we take the spectra of biosimilar medicine, and we check the similarity between the spectra. So if there is no difference between the spectra which is observed for reference and biosimilar, that means a total evidence is generated that the biosimilar and reference medicine is similar. The second is functional analysis, where we perform receptor binder studies. So here again, we plot the relative response of receptor binding affinities with the reference material and the biosimilar. And you all can see clearly over here that the results of reference and biosimilar are overlapping with each other. So again here, a total evidence is generated that the functional characteristics of biosimilar and reference medicine are the same. Then the pharmacodynamic data, again we check the pharmacodynamic studies, and again here the graphs are completely overlapping for reference and biosimilar. So again, there is enough evidence that the pharmacodynamic parameters of biosimilar and your biologics are the same. And again, efficacy and safety, again if you all see, the graphs or the curves of reference medicine and biosimilar are again overlapping. And this, when we perform all these studies in totality, we get enough evidence that the reference medicine and the biosimilar are similar in nature, and hence we can, and if we are able to get such types of results, then we can easily go forward with the biosimilar development. So the manufacturing process is quite simple, for saying purpose. But again, the first step is host cell line selection. Then we prepare a cell culture. Then we allow the cell culture to grow. The cell cultures are allowed to grow in size in large vessels. Then once we get the desired size of the cell culture, then we purify and we separate the desired material from that cell culture. So the purification is a critical step because though we have different methods for purification, still the purification step is the most lengthiest step so that we get the proper biologic material. So once we have purified the material by suitable techniques such as centrifugation or by chromatographic techniques or by filtration, we then have to go for the formulation. Okay, so by preparing suitable formulation, we can create a suitable dosage form for the administration of that particular biosimilar. So the manufacturing process for biosimilars and biologics is the same. The only difference is in the development process. So the approval of biosimilars, like biologics are already approved, but if we want to develop biosimilars, we again have to go through regulatory pathways. So the biosimilars are usually are approved by different regulatory authorities. For generics, as I told you, there is an abbreviated pathway which is given by the Hatch-Waxman Act, but that is not applicable to biologics and biosimilars. So the biosimilars, if we see the approval process in India, there are guidelines provided by the Central Drug Standard Control Organization, CDSCO, and these guidelines are developed by CDSCO in collaboration with DBT, that is Department of Biotechnology. And the guidelines were first, the draft guidelines were first prepared in the year 2012. Now, frankly speaking, India has the highest number of biosimilars which are approved. Around 90 biosimilars are approved in India. And the first biosimilar which was approved in India, it was for hepatitis B vaccine. So the guidelines in India are now in place. And these guidelines, they address the regulation of manufacturing process as well as quality, safety, and efficacy of similar biologics. So these similar biologics, which are biosimilars in India, they are regulated as per the DNC Act 1940 and Rules 1945. And they are also regulated under the Rules for Manufacture, Use, Import, Export, and Storage of Hazardous Microorganisms or Genetically Engineered Organisms or Cells 1989. And also it is notified under the EPA, that is Environmental Protection Act. These are some examples of biosimilars which are approved in India. These are a few examples. So for diabetes mellitus, insulin glargine was developed as a biologic, but now the biosimilar for that insulin glargine is available with the name Claritis. So for different indications, there are different biosimilars which are now approved in India. As I told you, the regulatory approvals, they happen country-wise. So different agencies, regulatory agencies of different companies, they are actively involved in the approval of biosimilars for those particular countries. So we have European Medicines Agency for the European Union, the United States Food and Drug Administration for the US, then Health Products and Food Branch of Health Canada for Canada, and so on. Also, World Health Organization has published its guidelines on the evaluation of similar biotherapeutic products in the year 2009. Okay, so WHO has provided this guideline so that the international in order to provide an international norm for evaluating biosimilars worldwide. So in summary, The biosimilar or a biosimilar drug, it is a medicine that is very close in structure and function to biologic medicine. So for some brand name biologics, there could be one or more biosimilars which are available. Okay, so one biologic and one biosimilar, this is not the case. For one biologic, there could be more than one biosimilars which are available. So the biosimilar, it has a structure which is highly similar, but not exactly the same to the brand name biologic. So the biosimilar, it needs to have no clinical meaningful differences between the biosimilar and the biologic. They could have some differences in the structure, but clinically they should have no differences. This means that the biosimilar is also considered as safe and effective as the biologic. So there is a worldwide evidence that today, due to the rigorous approval pathways of biosimilars, it can be stated that biosimilar is equally effective and safe as compared to the originator biologic. So that is why it can be safe for me to say that we can easily go for biosimilar. Biosimilars, they have been in clinical use for more than 10 years. And there is an evidence from more than 700 million patients worldwide. And it shows that approved biosimilars can be used as safely and effectively as their originator biologics. So the information that I provided to you all today, it was a very brief and overview of biologics. There is much more to it, but due to time constraints, we cannot go into much details. So thank you. Any questions?
So good morning, everyone. Good morning, everyone. I feel immense pleasure to take this opportunity to propose a vote of thanks to our eminent speaker, Macveena Machado, ma'am, who graced this webinar with the extremely relevant address on biosimilars. Ma'am, who graced this webinar with an extremely relevant address on biosimilars, ma'am, have rightly highlighted on various points like what are the definitions of biologics, what are the major differences, small molecules and the biological medicines. Also, what is the reference product? Why should we use the biosimilars product? What are the reverse engineering, stages of biosimilar development, and the totality of an evidence. So thank you all. We will be starting another session soon. Don't leave the meeting.